right, so now we are in 16.2. First, let's take a look at who's considered the father of epidemiology and why, and that is John Snow. He was a British physician, and keeping in mind, so this was in 1854 that this happened. Not long ago, in 1500 or so, late 1500s, is when people still thought of the miasma theory and felt that that was perhaps a reason for illness. It's just that there were these forces that provided illness and made some people sick, and that's just kind of the way it was. It was along the lines of kind of the gods are upset with us kind of thing. But this was still happening relatively close to this period of time. But John Snow, what he did was he instead determined the source of a cholera epidemic in London. And so this really, not necessarily solidified, but really started to make sense of the fact that diseases spread and can be transmitted and isn't just due to these miasmas or kind of this more fleeting, floaty kind of idea, but it was more concrete. So he investigated this in two ways. So what he noted was that there was an area of London where people were getting sick with cholera. And he wanted to figure out why it was that these people were getting sick with cholera and other people in London were not getting sick with cholera. So he investigated it. First, he identified that there is a water source for those that were infected. So he noticed, he went in and he interviewed all these people, and he found out that they all used the same water source. And this water source was a pump on Broad Street, is where this illness was occurring, or cholera was very common at the time. And downstream from London, downstream from this pump, the water was filled with garbage, and it contained sewage. People dumped their sewage there. So there was all of this garbage and sewage, and so he noted that all of the people on Broad Street here, near Broad Street, were all getting cholera, but they all had the same water source, and that water source had this garbage and sewage and dumping area upstream from it. So he noticed that is one thing. The second thing he noticed was that brewery workers did not get cholera. So even if they lived in that area, not everybody in that area was getting cholera. Some people were getting cholera and some were not, and he noted that brewery workers often did not get cholera. And he looked into that further and found out that brewery workers were provided with beer to drink while they were working, and so therefore they did not drink the water that was coming from that particular pump. And so he noted that not only did everybody use that pump in the area, but those that didn't use that pump were people that were not drinking water and instead were drinking beer because they worked at the brewery. So then what he did was he mapped these incidences. You can see his map here. He mapped the incidences of cholera. He noted that there's a high frequency among those that used a particular water pump on Broad Street. So then what he did was he went and removed the handle to that pump. So people could no longer pump water from that particular pump. They had to go to pumps elsewhere to get their water. And then what he noted was that the incidences of cholera went down. So this is the first time that somebody was able to go back to its source to find the etiology, to find the source of an outbreak. And not only did he do what epidemiologists do now, was that he went around, he investigated, he did analyses, he found the source of it, but then he also went so far as to have a response to that, have an effect to that. So he removed the pump handle, saw the incidences went down, and this is the first known public health response to an epidemic. So we had this epidemic, and he did something about it. This also led to shedding light on unsanitary sewage practices, right? So he was able to then directly correlate that to, look, we're dumping our garbage, dumping our sewage up above water, so it seems clear now that that sewage and water that we're going to be using don't mix well. So shed light on that and the effects of waste dumping in the River Thames. So keeping the river dirty was going to be affecting people's health, and people didn't like that. So then they started to pay closer attention to where they were dumping things. But then also, this also really shed light on the germ theory of disease, meaning that it isn't just, the gods are angry kind of thing, or there are these things in the air that are causing illness, but this was very specific, a fecal-oral route, meaning we're dumping sewage here, people are drinking the water that the sewage is being dumped in, and then they're dumped in and they are getting sick, so fecal-oral route, and noting that there are germs rather than these forces that are unseen that are making us sick. So again, John Snow is kind of then seen as the father of epidemiology. He's kind of the first epidemiologist. So let's take a look at the different types of spread. So we have two different types of spread, but the first one we're going to look at is common source spread. So common source spread is where we have a single source for all of the individuals infected. And so we see that in the example with John Snow and the Broad Street pump, is that we have a common source, right? So that single source was the pump. Now within common source spread, we have three different types of common source spread. The first is point source spread. So point source is where there's a common source that operates for a short period of time. So a short time period, often shorter than the incubation period for the pathogen. So that can make it really difficult to work your way back to it. So it's not like in the Broad Street pump where people got sick for a short period of time, or people got sick and then they didn't after that. In this case, the example that's given is a contaminated container of potato salad at a picnic. So if somebody has potato salad that's been sitting out all day during the picnic, it's contaminated, and then that goes away, right? Short time period. But all of the people that got sick by eating that one source. So that's a point source. The second type is continuous common source spread. So continuous, meaning that the infection occurs for an extended period of time. So oftentimes longer than the incubation period. So for example, the source of the London water. So this was continuously contaminated with the sewage, so people were continuously getting sick. So even though it was still a common source, there's one single source for everybody getting sick. This was continuous. It was continually being infected. The last is intermittent common source spread. So in this one is intermittent, as the name implies. So infections occur for a period, then they stop, and then they start again. So for an example here would be a well being contaminated after large rainfalls, and then that being cleared out. So we can oftentimes think of this as in if, let's say on a farm, a person has well water, say within an area, there are several farms, and they all use the same well, for example, and they have, they're keeping cattle and things like that. And then there's a huge rainfall, and then with that huge rainfall, we see an infection increase. That might be because that well is being contaminated with the feces or fecal bacteria from the cows, from the cattle. And then over time, that rainwater is no longer the rainwater. We haven't carried all of that bacteria into the well. And so then we see that go down. We see the causes of infection go down. And then maybe another big rainfall comes, and then we see another increase in infection. So this would be an intermittent common source spread. The second type of spread is propagated spread. So propagated spread means that it is through direct or indirect person-to-person contact. So again, this is in contrast to common source. So remember, common sources, we had one single source and people got sick from that one single source. We have our one single source, and then all of the people that are getting sick from this one single source. In propagated, we have, say, that single source, and then that gets somebody else sick, and that person gets other people sick, and this person got other people sick, and then that person that's sick now gets other people sick. So this is propagated. So in this case, we have no single source. Each individual then becomes a source for one or more infections. So unless this is stopped immediately, infections occur longer than the incubation period, right? So we don't have that point source where it's a very short period of time for infection, and then usually whatever the source is gone, like the potato salad's been thrown away long before people are seen at the hospital for E. coli or something. So point sources often lead to localized, large-scale outbreaks for very short time periods. So, for example, that contaminated well, that does happen, but oftentimes where we see this specifically is that where we have farmers that have cattle, for example, having their cattle be next to farms that have spinach, for example, or lettuce, or other leafy greens, for example. And then when we have this high amount of rain, it washes bacteria from the feces, into the spinach or lettuce fields, and then those don't get cleaned well enough before they're packaged up and sent out. And that's how we have some of these epidemics, or an outbreak from, say, spinach, for example, because it's bacteria coming from the feces of the cow due to a rainfall. So that would be a point source coming from one particular location, and this can lead to localized, so those that just ate from that particular area, or those that just get water from that particular well, for example. But they can be large-scale outbreaks, but it's for a short time period. It's not continuous. However, propagated spread often leads to our longer duration outbreaks because it's going from person to person to person to person, and we have to think about incubation times. So a person may think that they're better, or a person may not know that they're sick yet, and then they're still spreading that from person to person to person. So we often have these longer duration outbreaks. And they vary from small to large, so it might be a small area, but it can be large depending on the population. If it's somebody that's living in Detroit versus somebody living in somewhere that's very, very rural, or if it's somebody that's flying across the country or to another country, then it can spread and be much larger. Propagated spreads are then more difficult to stop, right? So our example with John Snow and the Broad Street pump, we can see that we take the handle off and the disease goes away. Or we clean up the river, the disease goes away. In the case of propagated spread, this is really difficult, because people are coughing and sneezing and, sharing food or drinks or whatever the case may be. So these are some images from the text. Here we have disease incidence with a point source spread and disease incidence with a propagated spread. So you can just kind of see this is just an illustrated version of what we were just talking about. So we often have a short period of time for this, a short duration for the illness and the number of cases. So the number of cases can be relatively high here, less, however, than the highest number of cases over here. But it's in a short period of time. We're looking at mostly just November here for the large number of cases, versus on the right here, when we have propagated spread, we have more extended duration. So you can see this going from November all the way into January is kind of where we see the high numbers, and even before that, in October. So we end up having more people with a particular illness for a longer period of time. So the next person we're going to talk about is Florence Nightingale. Florence Nightingale was a nurse. She was a British nurse, and she cared for wounded soldiers during the Crimean War. She really is seen as kind of the first person to develop the science of epidemiology because she did extensive note-taking and record-keeping. So she kept detailed notes on the causes of illness and death during the war. So previously, anybody that died from a particular illness, whether it be an illness from the wound itself or just, from being shot or something, so directly from the wound, versus my first one would be, for example, like actually getting hurt and then an infection being in the wound, or from just a generalized infection, not related to their wounds. So she kept detailed notes on all of those different types of deaths and illnesses. She ended up publishing a book in 1858, and what this did was it showed the monthly frequency data on the causes of death. So she kept such detailed notes that she was able to keep track of these things, and then she published that for people to know kind of what was going on. And what she determined was that most deaths were due to a preventable infectious diseases rather than the wounds that they were actually getting by being in the war. And so this was kind of, as I mentioned, she's the one that developed the science of epidemiology because she did this meticulous record keeping. She really spent time writing all these things down and taking meticulous notes so that this could be looked back on, so that they could really take a look at what was happening, so that then they could change this, because she was a nurse and she cared for these people and didn't like seeing all of these soldiers dying, but she noted that they weren't really dying from their wounds, more so they were dying from other diseases that were being transmitted. And then, therefore, like we see in epidemiology, that can be addressed somehow, depending on the practices and such, and hopefully those things can be stopped. So just some images from the text here. A here, this is Florence Nightingale, and then B, these are some of her notes, some of the stuff that was published, and it's just showing her fatalities here. So you can see that it mentions down here, people that died from their wounds were in pink. preventable infectious diseases were in gray, and then other causes were in brown here. So you can see that more people died from infectious diseases in the war than they did from their actual wounds. And so by presenting that information, then people can change things. The next person is Joseph Lister. And he was a physician during the mid-1800s, and he discovered the disinfecting properties of carbolic acid, also known as phenol. And you should already know about phenol and the different types of or variations of phenol. This provided early epidemiological evidence that led to good health practices in clinics and hospitals. So when he discovered the disinfecting properties of phenol, what he did was he demanded that people used it. So the people that he was around, he demanded that they used it. And therefore, what he did was he introduced these disinfecting protocols. So he determined that it had disinfecting properties, and so then he himself, as a surgeon, as a physician, decided he was going to use phenol to make sure that he wasn't infecting various people. So at this time, prior to Joseph Lister, if we can think back to that time, people used to just go house to house. This was oftentimes when doctors did house calls, they went house to house. And even if people went to a central location for something like a surgery, oftentimes people didn't clean surgical instruments between people. So they just didn't think of it at that time, right? If we can think about, the miasma theory, it's not because I took surgical tools that I was just cutting somebody's insides with and brought them to you and never cleaned them. It was just because of, you upset somebody. So back then, they weren't cleaning instruments between people. But Joseph Lister, he said, yes, this is something that we should do because he noted that there was a pattern to this. So he introduced disinfecting protocols that lowered post-surgical infection rates, so meaning that less people were getting sick from utilizing dirty instruments. So then he demanded their use during surgeries. So anybody that was around that he could tell, he then demanded that they use phenol to disinfect things between patients. So not only did he disinfect the tools and make others disinfect their tools, but also their hands, because again, at this time, they would be seeing many, many people, often going from house to house, and they weren't cleaning their hands. They weren't washing their hands at this time. So this was even during surgeries and things. So this became kind of commonplace at this time. So Joseph Lister was a big person in regards to making sure that things stayed clean and then not transmitting infection between persons. So some other types of studies. We're going to talk about ways that we can study these. So we went through some of the pioneers of epidemiology and talked about the different types of spread. Now what we're going to talk about are the different types of studies. So we said that we need to have not a simple association, but direct causation. So these are different ways that we can develop that or get to the direct causation. The first one is through an observational study. So this is how we are gaining information on a disease. So epidemiologists are going to be going out and gathering this information and producing these types of studies to try to get information about diseases and therefore hopefully affect them by preventing people from getting sick further. So an observational study is data that's gathered from study participants through measurements or answers to questions in interviews. So, for example, white blood cell count or questions about recent travel. So observational is just what is observed. So this would be if somebody goes around and they say, did you travel to Africa? Did you travel to Africa? Did you travel to Africa? Or wherever. Or they would go and get white blood cell counts from various people. So through these study participants. So people are going to be part of the study. As they are part of the study, they can either get measurements, meaning something like a white blood cell count or some other count like that, or just by asking them questions. So in this case, the subjects can be chosen at random from a population, and this population would be of affected people or unaffected people. So if we start to see some incidents of disease in a particular area, then epidemiologists may go to this area and then they're going to start asking questions. They'll start asking people where they've been or if they went to a particular function or at a particular time or asking questions about their health, things like that. In this case, the subjects are not manipulated by the researcher. So this is what we're saying here is that we're not gathering people and putting them in a laboratory or putting them in a clinical setting and then adjusting their health or taking different samples of things and then seeing how they change or how they're different or things like that. This is just observing what's already happening. This is easier to carry out compared to experimental studies, right? So it's just asking a bunch of people a bunch of questions or taking different samples, so it's easier to do. Sometimes this is the only option due to ethical reasons, right? So if we see an outbreak of something, let's go really crazy and say we have an outbreak of Ebola, we would not want to then like bring people into the lab and do an experimental study and say, oh, is this connected to the Ebola disease? You know, maybe we see signs and symptoms of Ebola and we want to test it out. We couldn't ethically test that out in a laboratory. So in that case, what we would do is an observational study, for example. Ask questions and then do lab results. So in this case, however, we're only able to measure associations between disease occurrence and possible causative agents. So this would not prove cause. Because again, we're just observing. We're saying, did you go to this place? Just because a person went to that place doesn't mean that there's that direct causation. Or just because a person has, say, a low white blood cell count, that doesn't mean that they have HIV or AIDS. They could have some other immunocompromised condition, or maybe they're just not feeling well, they haven't been eating well, you know, things like that. So it does not mean that there's this direct causation. So we can only measure associations this way. So we would need to have some other types of studies to prove the cause or to have direct causation. So some approaches in observational studies. We have descriptive epidemiology. So this is when we gather information about a disease outbreak, about affected individuals, and how the disease has spread over time. So this would be like an exploratory. This might be the first type of study that's done by an epidemiologist moving forward to determine what's going on with an outbreak. So in this case, we would have interviews with patients, interviews with their contacts. So the people that are actually sick, right? The patients ask them questions about their background, where they've been, who they were talking to, interactions they've had with others. Also with their contacts, so people that they contact, asking them if they have any signs or symptoms, or if they've gone to the same places or not, things like that. And their family members. So maybe family members can remember things a little bit differently than the patients themselves, especially if they're sick, you know, and their brain's not working very well, or their other contacts. So doing interviews is a way to approach an observational study. Also examination of samples and medical records. So taking a look at their medical records, meaning their background, their history, maybe history of their family. And then their samples, of course. Any samples that are taken in the laboratory, taking a look at that and comparing them to other samples that are taken. Also take histories of the patient, so histories of food and beverages that are consumed. So this would be a good thing to do, particularly if we're thinking of the example of the potato salad, for example. And then we might form the basis for developing a hypothesis of causation that could be tested by more rigorous observational and experimental studies. So again, if we do an observational study, in this case, by asking these questions and approaching it in this way, this is a way that we can kind of get some background information, give us kind of intro information, give us some simple associations, but again, not direct causation. So another approach in observational studies would be analytical epidemiology. So this uses carefully selected groups of individuals in an attempt to more convincingly evaluate hypotheses about potential causes for a disease outbreak. So in this example, we might start with the questions and things that we just talked about. And then after we've gathered that information, then we would use our carefully selected groups of individuals so that we can more closely evaluate the information that has already been gathered and any conclusions we might want to make from that information. So in this case, still the selection of cases are made at random, but it would be more carefully selected, meaning from this group of individuals or that group of individuals. The groups used to assist in teasing out associations with disease. So again, using these groups, we would hopefully try to kind of get more information as far as these associations. And different approaches would include things like cohort studies, case control studies, and cross-sectional studies, which we're going to talk about each of them. In this case, we're going to gather data by going back in time. Also, we can do retrospective studies, or as the events unfold forward in time, which are called prospective studies. So we can do what we had already spoken about, and do the asking of questions and things like that, gathering samples, and then as we move forward to try to take a look at these different hypotheses, a hypothesis is formed saying we think that this might be associated with this. Again, that's just an association. Then we can do other approaches, meaning these cohort studies, case control studies, cross-sectional studies, a retrospective study, or a prospective study. So let's take a look at some of these studies. So first, we have our retrospective studies. So this is when we gather data from the past, so retro, meaning past, retrospective, and what we're doing is we're looking for previous information on these present-day cases. So this is going to be used to examine associations between the factors that are chosen. So if researchers or epidemiologists have gone around and asked dozens of questions to all of these people, and they find out there are just a couple of questions that seem suspect, meaning that they might be the reason for the outbreak. Those would be the factors that are chosen. So they wouldn't ask all of the questions or look at all of those things. And they would look at the disease occurrence. So the associations between the factors that are chosen and the disease occurrence. So for example, medical history, something like age. So again, if things are affecting people of a particular age group or a particular gender or different occupational history, so if everybody works in the same field, things like that. So again, retrospective, saying what has happened in the past that could lead us to this present day issue, these present day cases. Then we have prospective studies. So prospective is where we then will follow individuals and then monitor their disease state throughout the course of the study. So in this case, we have data on characteristics of the study subjects that are taken at the beginning and then throughout the study. So we look at things as we see them now, and then what we're going to do is follow them as they move forward and see what changes, what is different, or what's the same. And then what can be done is those that become ill are compared with those that do not become ill. So we can examine these associations between the factors that are chosen and the disease occurrence. So again, and rather than in retrospective studies saying, okay, what did you do that's leading to this now? In this case, we're saying like, okay, you have this now, let's see what happens moving forward. And so then we would be gaining different information based on whether or not we're using a retrospective study or a prospective study. The next type of study is a cohort study. So our cohort studies are going to examine groups of individuals who share a particular characteristic. So for example, those that were born in the same year at the same place, or those who practice or avoid a particular behavior. So for example, smokers versus non-smokers, right? So if they're looking at a particular group, we're examining a group of individuals that share a particular characteristic. We're saying, okay, we're going to compare this illness that we see in smokers compared to an illness that we see in non-smokers, for example. So it's usually based on characteristics or characteristic subjects that have been exposed, characteristics that subjects have been exposed to through their own choices or circumstances. So again, in this case, it's not an experimental study where we're saying, like, now we're going to expose you to something. In a cohort study, typically what we're doing is we're taking a look at what they're already exposing themselves to, and then seeing what happens because of that. So we're taking people that are smokers and either looking at their past to see how they are now. So because this can be either retrospective or prospective, saying, okay, for the example of smokers, we can say, you have this particular disease state now, what have you done in the past? Like, how long have you smoked, for example? Or what kind of cigarette do you smoke? Or, that kind of thing, which would be a retrospective study. Or what we can do is we can say, okay, you're a smoker, you're of this age, smoking this thing, and then we can say, let's take a look at you for the next 5 to 10 years. If they quit, fantastic. If they don't, then we have that information. We're not forcing them to continue smoking, for example. That would be a prospective study. So when we do cohort studies, we can have a single cohort. single cohort. And that is when we have those that are infected compared with those that are unaffected in the same group. So we can take a look at smokers, for example, and we can see smokers that are infected with some particular thing. Maybe they have a particular sign or particular symptom. And then we can compare that with those that are unaffected in the same group. So again, smokers, but they don't show that particular sign or symptom of had that infection. So then these outcomes are recorded and analyzed so that we can identify these correlations between the characteristics of subjects in the cohort and the disease incidence. So again, we analyze these things, we record them, we say we see this is the information we've gathered either retrospectively or prospectively, and then this is going to be useful to determine the cause, and this is without exposing subjects to risk, right, or bringing up ethical concerns, right? So the example of smoking is a good one because we're saying, like, we're not forcing people to smoke. We're just saying, like, of you people that are smoking, we can either look in the past or look in the future. And if you change that behavior, that's fine. Then you're just no longer in the study. So we're not exposing them to further risk than they're already exposing themselves or violating ethical concerns. The next one is our case-controlled studies. So this is typically retrospective, so looking into the past, and this is where we compare groups with a disease to those that are similar, but they don't have the disease. So this is a little bit more efficient than cohort studies. What we do is we deliberately select subjects who are already infected, and then compare that to people that are waiting for infection to occur. So people that we feel like have been exposed to something, so people that we know have been exposed. So we deliberately select people that have already been infected, been exposed to something and are affected, and then those that have been exposed to something and we're just waiting to see if infection occurs or when infection occurs. So typically retrospective, meaning that we look in the past and we say, okay, all of these people were at the same place at the same time or whatever the case is, and then we can compare those that currently have the disease to those that were at the same place at the same time but don't have the disease, and we can see if they developed a disease or if they don't. The next type is a cross-sectional study. So this compares randomly selected individuals that are affected by a disease or condition to randomly selected individuals that are unaffected at a single point in time. So in this case, we're saying all of these random people that are affected by something, random people that are not affected at one single point. So it's not a retrospective study saying, like, let's look at what happened over the course of time and what got you here. It's not prospective in that we're not saying, like, let's take a look at this as it moves forward. We're saying at this particular point in time, on this day, at this place, we take a randomly selected group, look at their disease state, randomly select a group of unaffected people, and then we compare them. So we compare to look for associations between different variables and the disease or condition. So at that day, at that time, what is it that makes diseased people different from not diseased people? This is also useful in determining the prevalence of a condition. So again, taking a look at a certain day, at a certain time, we can figure out all the people that are diseased, and then we can use that information for the prevalence of a condition, right? So how many people, remember prevalence is the number of affected people, so morbidity, at a particular time, at a particular single point in time. So that helps us determine prevalence, and then we take a randomly selected group of those people and compare them to unaffected people at that same point in time. The next one is experimental epidemiology. So rather than our observational, now we're taking a look at experimental. So what we were looking at, again, is observational. We were making observations. We were looking at what happened, what will happen, what is happening, and comparing with these different groups. Now what we're going to talk about is experimental epidemiology. This doesn't happen as much. We have a lot of other observational studies because of ethical concerns. So experimental epidemiology uses laboratory or clinical studies in which the researcher manipulates the subjects to study the connections between diseases and potential causative agents or to assess treatments. So for example, the administration of a drug. So this is on a volunteer basis, if we can get past the ethical concerns here, and that is all depending on the type of disease, so the type of drug, the side effects of the drug, things like that, which are all going to have to be presented to the FDA and things like that, and pass all of these checks and balances before it can actually be done. But when it does, we get to the administration of a drug, for example. So if somebody enrolls themselves in a study because they have a particular condition, then they can enroll themselves in the study. This is an experimental epidemiology study, where then we can administer a drug and see if they get better or see if they don't. Another example is inclusion or exclusion of different dietary items. So again, we're manipulating, the researcher's manipulating, saying, okay, we're going to remove this from your diet, or we're going to add this to your diet, and we're going to see what changes. Also physical exercise, so increase or decrease, stay the same, or a particular surgical procedure. So if somebody, again, this is if somebody's already signing up for this, they can then opt into a procedure or not. These experimental studies use animals or humans as test subjects. So a lot of times animals are used because we have ethical concerns with actually providing somebody with a particular illness to see what signs and symptoms are showing. So that would be on animals, but some of these examples up here would be useful for humans. They're more difficult to do and sometimes impossible, again, for ethical reasons. So observational studies are much easier to come by. Experimental, not as much. They do, however, provide strong evidence supporting a cause. So remember, in this case, we can get a lot of direct causation rather than our simple association. So other factors are held constant in an experimental epidemiology study. And then the only change is providing the drug versus a placebo, for example, if we're talking about administration of a drug, or having someone continue with a regular behavior versus adding something to their diet or increasing exercise or whatever the case may be. It is important, however, to understand the different types of studies, experimental designs, when you read articles. And we're not going to go into too much detail here, but it is important that the studies be blind studies, particularly double-blind studies. And that's because in a double-blind study, neither the subjects nor the researchers know who gets the drug and who gets the placebo. And this is important because it's going to remove bias. So if you can imagine, if we went to a group of people and we said, okay, your group of people, you are going to get this experimental drug that's going to cure your illness. And we went to another group and said, you're going to get the placebo, and we just want to see how your illness progresses compared to those that we give the actual medication to. This would then cause bias, for sure. The people that got the pill, let's say the drug, might notice a lot more often that their illness got better because they already in their mind are thinking, well, this is going to help me, and so then they'll see these changes or these improvements, versus the placebo group knowing that they didn't get the drug, then might not report things that are getting better. They might not get better because they already are expecting to not get better. So that level of bias, that we want to remove that. So oftentimes that's pretty self-explanatory, but not doing that is important. So that would be a blind case study or a blind study, is not telling the patients, not telling the subjects. Now, a double blind means that even those that are administering the drug in this example also don't know who's getting the drug and who's not. It's removed even further. So in this case, if we're just talking about a blind study where the subjects don't know, we might have 20 people in a room, 10 get the drug, 10 don't. And if it's a blind study, then the researchers still know who's getting the drug and who's not. So they might walk over to a person, and as they give the person that particular drug or placebo, they know who's getting the drug or who's getting the placebo. This can still cause bias because as they're taking information from that person might say something like, well, I did notice that I didn't have, that I slept more than 5 hours last night. And the researcher might be like, oh, that's because you got the drug. Okay. Versus if they don't know, then they wouldn't have that bias. They might just write down something like, got more than 5 hours sleep last night. So it's important for it to be a double-blind study. Double-blind means that even the person that's taking the notes from the person that is reporting back, or the subject, or is providing the drug or the placebo, they don't know as well. So it's still one step removed. And that is done by having researchers at another level that are saying, okay, these little cups number 1 through, or even numbers all have the placebo, odd numbers all have the drug, and they know that, but then they put all of the numbers on the table. Then the people that are actually administering the drug are going to come in when the other people have left, the ones that know that even and odd are different. Then they come in and they just take the numbers for their people, their subjects that they are administering the medication to or placebo to. So they don't know. And this is called a double-blind study. And so this is really important because it removes the placebo effect. So a placebo effect is the knowledge of treatment by the subject or the researcher and how that affects the outcome. So a placebo effect is as I was describing, where a person might think they're feeling better because they know that they're getting the drug or not. And so then that would affect the outcome. And we don't want that to happen. So as you're reading scientific literature, make sure that you're looking out for those double-blind studies because those are the ones that are going to provide you with the best information.